Okay, thank you, ma'am. Now we move on to our next lecture by Professor Varsha Gupta from the Department of Life Sciences, CSMU Kanpur. She's completed her PhD in Life Sciences from Jawaharlal Nehru University, New Delhi, and postdoctoral training in structural and functional genomics from the National Center of Plant Genomic Research, New Delhi. She is a recipient of several awards, such as National Fellowship from the Department of Biotechnology, the DBT, Government of India, during her MSc. She has also received JNU UGC fellowships such as JRF and SRF, as well as the Young Scientist Fellowship from the Department of Science and Technology Award for scientific achievements on International Women's Day by Rajashri Foundation. She also serves as a reviewer for international journals like Acta Physioplantarum, Current Orthopedics Practice, and Plant Physiology. She has received great recognition and funding for her research projects by the Government of India. Now I shift it over to you, ma'am.
Thank you, Siddharth, for the nice introduction. I think we'll have the recorded version played. Thank you to all ICB ILS team. Dr. Asmita Das, can we have the recorded version, or I can also present if you say? Okay.
Whatever seems fit could be done. Should I just start
Yeah,
this
you
one?
can play this. Yeah.
All right, ma'am.
Yes,
Hello,
actually,
everyone.
watch this,
Good
and
morning.
I think there is some issue with the connectivity, so it's better to play the recording, I guess.
Yes. First of all, I would like to thank the organizing committee of International Conference on Innovations in Biotechnology and Life Sciences, and especially to Dr. Asmita Das for giving me an opportunity to speak in front of all of you in this conference. So I would be talking about life with COVID-19, protective measures and challenges. As we all know that in December 2019, there was outbreak of pneumonia of unknown etiology in Wuhan, Hubei province, China. And this outbreak of pneumonia, like symptoms, it basically was related to severe acute respiratory syndrome coronavirus 2, that was also known as SARS-CoV-2, and the virus was believed to be have originated in the bats, and then it was transmitted to the humans, and then human to human transmission occurred at a very fast speed, at a rapid rate. Because of that, World Health Organization in the month of March, 11th March, it has declared COVID-19 as pandemic. Of the data from WHO 17 December 2020, there have been 7 crore 28 lakhs 51,747 confirmed cases of COVID-19, with 16 lakh 43,339 deaths. Now this virus is infecting at a very high rate, and it's a new beta coronavirus, which is severe acute respiratory syndrome coronavirus 2, that is SARS-CoV-2, and it is a seventh member, and it shares nearly 80% of its sequence homology with acute respiratory syndrome coronavirus and Middle East respiratory coronavirus. These are some of the episodes of this family of viruses which have affected the human beings, and majority of them they have been transmitted from the animal. So these are all zoonotic viruses. So in 2020, we are evidencing the COVID-19. Because of that, nationwide lockdown they have been imposed in several countries. SARS-CoV-2 and SARS-CoV, they highly resemble with each other, including MERS-CoV. So SARS-CoV-2 originated in 2019, SARS-CoV in 2002, MERS-CoV in 2012. So these two have originated from China. This is in Middle East. The host receptor to which they bind is angiotensin converting enzyme 2. SARS-CoV as well binds to ACE2, and MERS-CoV has a separate receptor. And the blood test reveals different symptoms in the blood. Though the mortality case fatality rate of SARS-CoV-2, as we can see here, is quite low as compared to SARS-CoV and MERS-CoV, but the virus is highly contagious. Because of that, there is a very high rate of transmission of this virus. So the coronavirus family is so named because of the presence of large spike protein molecules on the surface of the virus, and because of this spike protein, it gives a crown-like shape. So the virus is known as coronavirus. The coronavirus genomes are the largest among RNA viruses. So family has been classified into at least 3 primary genera, alpha, beta and gamma, and 7 viruses are currently known. Of these, NL63 and 229E, they belong to alpha genus. OC43, this was responsible for Russian flu. OC43, HKU1, SARS-CoV, MERS-CoV and SARS-CoV-2, they belong to beta genus of this family. SARS-CoV associated coronaviruses, they are family of single-stranded enveloped RNA viruses, and their genome have a number of open reading frames ranging from 6 to 11. They have a positive RNA strand, and the first ORFs, they contain most of the viral genome and encode 16 non-structural protein, while other ORFs, they encode structural and accessory protein. An entry of the SARS-CoV into the host cell is mediated by its interaction, the viral spike protein's interaction with the host receptor, which is angiotensin converting enzyme 2 receptor, that is ACE2, and the SARS-CoV-2 shows very high sequence homology with SARS-CoV to receptor binding domain structure, and this new virus could also bind ACE2 as we discussed in the previous slide. And the spike protein is functionally made up of 2 subunits, S1 and S2. Now SARS-2S, it uses ACE2 for entry, then protein S is modified by TMPR SS2, that is transmembrane proteinase serine 2, and after this modification, the entry of the virus particle into the cell is facilitated. So the former mediates viral attachment and the later favors the fusion of the 2 cell membranes, leading to entry of the coronavirus. The affinity of CoV2 with ACE2 is very high. It is 10 to 20 times stronger than its SARS-CoV, the previous virus SARS-CoV. So because of this, the infection of SARS-CoV-2 is occurring at a very rapid rate, and its transmission rate is very high. And this ACE2 receptors are expressed on lungs, apart from the respiratory tract, upper and lower respiratory tract, lung 82 cells, upper esophagus, and absorptive enterocyte from ileum and colon. And these also are co-expressed, both ACE2 and TMPRSS2, in esophagus and lungs, leading to the entry of virus from all these sites. Now this is basically it's zoonotic virus, so many animal reservoirs are proposed to be, so different animal reservoirs, they are believed to be the reservoir of this virus: pangolin, snakes, bat, pig and dog. So it can manifest, its infection can manifest systemic infections. It also can involve heart symptom, in which acute cardiac injury and cardiac arrest. We now listen to many people death because of cardiac arrest, kidney dysfunction, intestinal, circulatory and respiratory. And SARS-CoV-2, the infection can be asymptomatic or mild to severe pneumonia, and patients with COVID-19 disease, they can also experience the immune system problems like higher leukocyte numbers, higher levels of plasma pro-inflammatory cytokines, leukopenia, and highly upregulated pro-inflammatory cytokines in the patient's body. Now again, in this slide we can see that transmission can occur from human to animals, from human to human as well. For prevention, there are various guidelines which are being issued by government from time to time, and in the case of pneumonia, phlegm, breathlessness, cough, fever and chest pain, we should regularly, if these symptoms are there, we should consult the doctors. Children, smokers and old people are at higher risk for this COVID-19 infection, and the people who are comorbid for other diseases, they are also at high risk for COVID-19. So the typical symptoms of coronavirus infection, as we discussed, they are a fever, dyspnea, headache, cough, and myalgia. So these are the number of patients which manifest these symptoms. Now, our understanding of the coronavirus is evolving, and as it is growing up, we see that many patients are asymptomatic and many patients they suffer from the coronavirus disease 2019 with a respiratory clinical manifestation, and some of them they also experience multiple organ failures which lead to death in several cases. And less common symptoms associated with COVID-19 includes headache, hemoptysis, nausea, vomiting, and diarrhea. Because the pandemic is going on, so our understanding of SARS human to human transmission or human to animal transmission is still evolving. But air droplets, fecal, oral spread, and airborne transmission may be important sources of transmission. And all of us are evidencing the nationwide lockdown. So the virus has destabilized health system, economies, and governments around the world, and many people have lost their jobs because of this pandemic. So protection from this virus is the key to protect yourself from the infection and the clinical manifestations. So washing hands and feet, and whenever if somebody is moving outside the home, use of alcohol based sanitizer or hand rub is advisable. Virus doesn't travel through the mobile network, so there was a myth that was clarified, but virus can travel with the items which we purchase from outside. It can sit on various items, so we have to take care whenever we are purchasing or getting things from outside. Social distancing and mask usage should be followed, and fraudulent COVID-19 tests, vaccine, and treatments which are present in the social media and social groups, they should be avoided. Only the regular or the authorized website should be visited for any kind of information related to COVID-19. Now, the virus can spread from an infected person's mouth or nose in the small liquid particles when they cough, sneeze, speak, sing, or breathe heavily. And these liquid particles they are of different sizes ranging from larger respiratory droplets to small aerosols. Now if some other people catch these droplets through their mouth, nose, or eyes, they are and the distance is less, then they are likely to contract the virus. So this is how the virus contract. Aerosol transmission occurs more when we are in indoor settings in which crowded and inadequate ventilation is present. So there in those places there is more chances of contracting the virus. So we should avoid now we should avoid going to the places which are highly crowded, which don't have sufficient ventilation, and we should not stay at these places for longer duration. We shall talk about this later. So this is what we can see. So after sneezing or loud speaking, these are the droplets. So larger drops, they are falling down, but as you can see here, the smaller drops, they are traveling to a they can travel the great distances. So this is almost 10 feet. So let's say a bus is going and the people are walking on the road and they are thinking that they are walking alone. But if somebody sneezes, some COVID-19 patients sneezes from the bus, then they are likely to contract the virus, though the dose of the virus will be lesser because it is open area. The viral load is important for infection, because if we are in the indoor settings and we contract the virus and the load of the virus is higher, then the problems can be very severe. That's what we are evidencing in the clinical, the doctors who are handling the patients, because they are in COVID wards and they are continuously handling the patients. So though their age and their immune system and other factors may not be responsible for the demise of many doctors, we have lost many doctors, many medical practitioners in this COVID-19, but that is because of acute viral load which the body is handling, and this viral load the immune system is not able to clear. Now after coming back from office or from work, everybody should do gargling with lukewarm saline water for protection of nasopharynx, oropharynx, and posterior part of nasal tract. In a German study, it has been proved that this tract is a reservoir for the virus, and is responsible for transmission. So gargling with lukewarm saline water will be helpful. Mask is protective, so it should protect from contamination of nasal tract and oral cavity. Masks should not be N95 masks, they should not be washed, these masks should be continuously worn if the people are going outdoors, and if they are wearing cloth mask or lesser than N95 mask. So please remember that some of them are there to check the transmission of the virus from you, and some of them are protective for you to contract virus from other people. So when going in very crowded setting, make sure that people are wearing N95 mask. Not to be loud while speaking. Now the winters have arrived, so precautions for clothing, because in summers we were washing all the clothes, but in winter we won't be able to wash the sweaters and all. So clothings can be hung outside on the terrace in sunlight, or they can be we can spray the sanitizer, not the hypochlorite but the sanitizer, and then don't wear it again for, let's say, 2 to 3 days. Then we can wear our sweaters, coats, or other warm clothings after that. A flickering of clothing or protective covering should not be practiced. And COVID-19 can spread from, as we talk, person to person. It can also spread from different devices. Now this is a systemic respiratory disorder, and the incubation of COVID-19 is 5.2 days. So nearly within this time period, the patient is asymptomatic. So in this 5.2 days, the virus is there, the virus is multiplying, but the patient is asymptomatic, and this is the time when this patient is basically transmitting the infection to many other people nearby. So there are a variety of clinical manifestations which we have seen with COVID-19. Now infected people, as we discussed, they can be contagious and the virus can spread from them to other people. Now infected people appear to be most infectious just before they develop symptoms, namely 2 days before they develop symptoms. So 5.2 is the incubation time. So before the clinical manifestations of COVID-19 they appear, they are most infectious, and since they are asymptomatic, so people will not really understand that they are suffering from COVID infection. So the best precaution is that take care and think that everybody is suffering from COVID-19. So keep your protections on. So someone who never develops symptom can pass the virus to others. So that is the basic risk. And the things which we should avoid is crowded places, close contact settings, confined and enclosed spaces. These should be strictly avoided. So just a small difference between pre-symptomatic and asymptomatic. So asymptomatic are those who have contracted the virus but they are not presenting the symptom. So they have the virus but the symptoms are not seen. But pre-symptomatic patients they have contracted the virus, the clinical symptoms are going to come, but this is the stage which is before the appearance of clinical symptoms. So again, the closed setting should be avoided, and if there is all the three things, crowded place, close contact setting, confined and enclosed spaces, the risk becomes very, very high. So government is continuously issuing guidelines for people, so that should be strictly followed. So social crowded place, maintaining social distancing, keeping windows, doors open, keeping hands and mouth clean, wearing mask when moving out is important. And whenever people are traveling, they should follow local guidance. Whatever guidance is coming from national, regional or local authorities, they should be followed. And while attending any kind of program or function, before attending that, you should weigh how many time, for how long you would be there in the program, how many people would be there. It will be close contact setting or it will be open program. So all those things should be taken care of while attending any kind of function. Even co-infections have been reported to occur with COVID-19. Many of the patients, they have reported the co-infection, but many patients with COVID-19, they have reported the appearance and occurrence of co-infections. So these all things should be taken care of. Now diarrhea is another frequent symptom which is associated with coronavirus, And in the SARS-CoV-2, it has a prolonged duration in GIT rather than in respiratory system. So there is high possibility that the stool of infected children, it can be responsible for viral transmission when the schools and kindergartens they are open. So parents and children should be trained accordingly for these all things. And if somebody at home is suffering from coronavirus, then take care that others are not using the toilet which a COVID-19 patient is using. Now with asymptomatic and younger age, there is lot of anxiety, there is lot of problems which we are also facing, our students are also facing. So these are all disturbing the people mental equilibrium, economy, employment, and they have affected many things. We have developed unknown fear of the virus. So these all things, they are happening in the population. So careful monitoring of epidemiological behavior of COVID-19 and the clinical data, the vaccines and all, these should be properly weighed and care should be exercised. Now shoes and all should be they should be outside the house. The COVID-19, as we discussed, is related with major psychosocial effects. So many people during the lockdown, they have faced a lot of problem, lot of psychological problems. Many of the people they have experienced. COVID-19 has brought a lot of unknown risk. Many people have succumbed to COVID, risk of anxiety, risk. All the time we are under psychological pressure. Some people are experiencing sleeping problems as well. Many people we have seen, they have succumbed to COVID. So that have led to a lot of stress, lot of pressure on the minds of the people. Anxiety, so people are living in anxiety, their daily life is affected. The people who are daily wagers or daily workers, daily wage workers, their life is also affected because of COVID-19. So this is one important aspect which should be taken care of. And best part is that every person should be considered as suffering from COVID-19, so that all the preventive measures, all the preventive things should be in place. So because people who are asymptomatic, they behave just like the normal people. So, and this is the slide whereby I'm trying to show that people are thinking that once they have contracted COVID, so they will not be reinfected with COVID. So this is viral reassortment, recombination and mutation. In all the three conditions, there can be shifting, there can be genetic drift and antigenic drift. Because of that, if once they have contracted the COVID-19 virus, there is a possibility that they can be infected with virus again. In fact, some of the people who were infected with virus, they are reporting reinfections. That is the thing which people should take care of. And with COVID-19, death is not the only adverse outcome, as we know that many people are suffering and experiencing prolonged, fluctuating, debilitating symptoms even several months after the initial mild disease. Some of the people who have recovered, they experience symptoms after some time. So these all things, they are very important. And in this, the people who are associated with hospitals, the doctors, nurse, it is essentially important for the medical practitioners to take care. And for other people also, it is very important to do exercise regularly, to do breathing exercises regularly to avoid the symptoms of COVID-19 virus who have contracted the virus. So all the WHO and government guidelines, they should be strictly followed. Now coming to vaccines for COVID-19, as we all know that vaccine development program, it requires time. So traditional vaccine development, it's a lengthy process and it requires nearly 15 years. And when the vaccine is developed, the process usually begins with the vaccine designing, evaluation in animal models. After that, in which the formal after that in formal preclinical experiments they are conducted, and the process for vaccine production is designed and toxicological studies are performed, and this stage can also last for several years. Then an application for an investigational new drug this is filed. So after this investigational new drug application is filed, and phase 1 clinical trials they will start. So phase 1 clinical trial, they will involve lesser than 100 individuals, and they are performed, and in this initial safety profile of the vaccine candidate is checked and immunogenicity data is obtained. If the results are promising and good, then it is moved to phase 2 clinical trial. Then in phase 2 clinical trial, testing is done in few hundreds individuals. It also takes time, and then further immunogenicity and appropriate dose, optimal vaccine regimes, they are all finalized, and then it moves forward with phase 3 clinical trials, in which thousands of individuals are involved, and if efficacy and safety is evaluated, and if the outcome of phase 3 trial meet the predefined endpoints, then a biologic license application, that is BLA. So after phase 1, phase 2 and phase 3, this biologics license application is filed, and after that large scale production of the vaccine it starts. Now this is the traditional method of production of the vaccine, but in the case of COVID-19, as we have seen that rapid action and the development of vaccine that has occurred at unimaginable unimaginable time frame. Okay, so SARS-CoV and MERS-CoV vaccination programs, they have enabled the initial step of exploratory vaccine designing to be essentially omitted in the case of COVID-19, which has saved considerable amount of time. Now, in many cases, the production processes for vaccines, they were adapted from existing vaccine candidates, and in certain cases, preclinical and toxicological data from related vaccine trials they were used. Now several manufacturers have already started the commercial production of vaccines at risk without any result from phase 3 trials. Now the license are not yet clear, but it can be some vaccines may be approved through an emergency use authorization. So we all are hearing that few vaccines are going to come to the market. So here, as we can see, the process is really as compared to the traditional vaccine designing, it is almost 10 months to 1.5 years to develop a vaccine for COVID-19 with phase 1, phase 2 and phase 3 clinical trials. Now vaccines against SARS-CoV-2. So what vaccines can target SARS-CoV-2? For this, it is important to understand the structure of SARS-CoV-2. So SARS-CoV-2 is as we can see, it's the vaccine can be targeted for its spike proteins. So this is spike protein, this is membrane protein, this is small envelope protein, or it can target its RNA or the enzymes required for its other work. Was quick sequencing of the viral genome, and after this phase 1 trial they were initiated in March itself. There are nearly 180 vaccines at various stages of development, in which nearly 50 they have gone forward, and data from phase 2, phase 1, phase 2 and phase 3 clinical trials is getting available for some of them. And the important protein for COVID-19 is its spike protein, and in the spike protein also its receptor binding domain is important for binding with this ACE2 receptor. So this has been targeted as a potential vaccine candidate in the case of COVID-19. Then antibodies, because it is seen that neutralizing antibodies against this spike protein, they can prevent its attachment to the host cell and neutralize the virus. So a spike protein was identified as an important antigenic target for developing vaccine against SARS-CoV-2, but there are other candidates as well. So this is a receptor binding domain of the spike protein. Now this can be used as a potential vaccine candidate. So this will be a subunit vaccine, but the problem is the receptor binding domain is that the protein is very small and it lacks many other spike proteins, so is not able to completely present the virus to the host immune system. Then inactivated vaccines that contain SARS-CoV-2, that is grown in cell culture and then chemically inactivated. Live attenuated vaccine, in which genetically weakened or attenuated strains of COVID-19 are used, but it takes long time to develop. So codon optimization can be used for this kind of vaccine. Then recombinant spike protein based vaccine, in which recombinant spike protein is cloned in some of the already used viral vectors. Replication competent vector vaccine,
It's
they
12
can
o'clock.
propagate to some extent in the cells of vaccinated individual. And then inactivated virus vector vaccine, they carry copies of the spike protein. And DNA vaccines, they consist of plasmid DNA. RNA vaccine consisting of RNA encoding the spike protein and are typically packaged in nanoparticles. Now these RNA vaccines, they are basically developed by Pfizer and Moderna. So the vaccines which we are hearing these days, they are coming from Pfizer and Moderna, so that we shall see. And another from the Serum Institute of India, this is CHADOX1N CoV-19, that is basically University of Oxford AstraZeneca and the Serum Institute of India, and it is based on replication incompetent chimpanzee adenovirus expressing a wild type version of the spike protein. So this is without any stabilizing mutations, and this vaccine candidate was tested in rhesus macaques intramuscularly, and both developed neutralizing antibody responses for this vaccine. So antibodies against the spike protein that target the RBD, receptor binding domain, and other regions of the protein can neutralize the virus. But antibody response to the spike protein is highly varied, in which initial boost is observed with reduction and then stabilization of the response. Now when the natural infection occurs, the virus usually enters through the respiratory tract, leading to mucosal as well as systemic immune responses. Now mucosal immune response leads to the production of secretory IgA, while the systemic immune response leads to the production of immunoglobulin G. When there is transdermal or transmuscular injection for the vaccine, it only leads to the production of IgG, while in upper respiratory tract the major neutralizing antibody present is IgA. So that is also very important, because we require both the responses of both the antibodies IgA and IgG, but the mucosal antibody response, they will not be present if the vaccine is injected intramuscular or intradermal, and in upper respiratory tract the major antibody is IgA. So it will be difficult for the antibody to check the virus once it is entering in the body. In non-human primates, infections with SARS-CoV, they have shown to protect against reinfection and is primarily mediated by neutralizing antibodies. So these intradermal intramuscular vaccines, they have their own limitations. Now there are several vaccines which are getting developed, but these three from Pfizer, Moderna and the Serum Institute of India. Of these three, Pfizer vaccine showed 95% efficacy, and it applied for EUA 2 days later, and the Britain becomes first country in the world to approve the vaccine. Now Moderna vaccine has also applied for EUA approval in the US, but for Serum Institute of India, the trials were halted after adverse events in the UK. But in November, its phase 2 trial showed a good immune response and an average efficacy of 70%. Still waiting for this vaccine to be successful. So hopefully we'll have vaccine soon. COVID-19, there are currently no specific medication, but still there are some of them which were being used. So we have heard about hydroxychloroquine and chloroquine. Now these are antimalarial drugs, and they have showed good protection. They have been used in long-term treatment in rheumatology without significant side effects. So actually these chloroquine and hydroxychloroquine, they change the pH of the cell membrane surface, and they inhibit the fusion of the virus with the cell membrane. They also inhibit nucleic acid replication, glycosylation of the viral protein, assembly of the virus, and release of the virus from the infected cell. So in vitro's activity against SARS-CoV-2 has shown protective results, but the clinical studies in humans, whether it can completely prevent the viral entry and all, they still need more time. Chemostat is another important drug which inhibits TMPRSS2 protease, so basically prevents the fusion of the virus with the cell membrane. Remdesivir is antiviral for Ebola, inhibits RNA dependent RNA polymerase, prematurely blocking RNA transcription. So remdesivir came up as a very good drug against the COVID infection, and the safety profile has been demonstrated in Ebola studies. So this has shown very good effects in the severe infection with new SARS-CoV-2 infections, and emergency use authorization has also been provided. Lopinavir, ritonavir, LPV, RTV, they're also antivirals. So this is another alternative drug for COVID-19, and this is a protease inhibitor used to treat HIV infection in combination with ritonavir to increase its availability. Then there are certain others antiviral agents which are getting used. Tocilizumab, this is IL-6 receptor antagonist. This is also proving very important in the case of COVID-19 infections. As we can see here that this is angiotensin, this is TMPRSS2. So how the viral entry is there, and there are there are drug candidates which are acting at different phases, but till now no drug has been documented to have complete protection from the COVID-19, though there are several drug candidates which are tested for COVID-19. So we have already talked about many of them. So best remedy for COVID-19 till now is that we should prevent the virus from entering our body, and after it has entered our body and we are suffering from COVID-19 infection, so we should get ourselves thoroughly checked. We should exercise all the precautions and preventive measures whatever are being suggested by the government. And nowadays what is happening is people are not at all bothered about COVID-19 infections, and they are thinking that COVID-19 is gone. But every day many people are lost because of COVID-19 infections. So we should not exercise relaxation. We should not exercise the kind of life which we were having before COVID-19. So the best key for COVID-19 is till the vaccines are tested. Even if you take vaccine, it is important that after vaccination also we should maintain or we should exercise the regular routine preventive measures which are given by the governments. We should follow all the guidelines of our government and the WHO, because the COVID-19 virus is mutatable. So vaccine may not be, it will be good, but vaccine may not be effective against all the viral strains. So we should strictly follow government guidelines. So जब तक कोई दवाई नहीं तब तक कोई ढिलाई नहीं. So living with COVID means living with precautions. Keep yourself healthy, active and safe. Once again, I thank the organizing team, Dr. Asmita Das and whole DTU team for giving me an opportunity to present my talk, to present this COVID-19 thing in front of you. Thank you so much.
Thank you, ma'am. This is was a really great session.
Thank you.
Yes, ma'am. Now we have a few queries which I'd like you to answer, ma'am. Ma'am, the question is, what precautionary measures should be taken by the individual who are more susceptible to common cold problems, and how we differentiate whether it's due to a COVID infection or a normal common cold?
Okay, for the first part, the people who are more prone for common cold issues, they should take all the precautions for countering the virus. Whatever guidelines are being given by the government, they should follow. And second part is, for people who are contracting cold regularly and the people who are normal, both of them should take precautions. And for differentiation of COVID with normal cold, COVID usually comes with dry cough. It's not running nose, is not a very typical symptom of COVID. So people who are with running nose, then probably it is not COVID. Otherwise, majority of the symptoms they overlap, but COVID is with a little bit different symptoms as compared to the normal cold.
Okay, thank you, ma'am. And ma'am, another question states that what are the chances of reinfection of COVID-19?
See, and not everybody gets reinfection, but some people see your immune system is highly protective against COVID 19. And if you see the Pfizer's success rate, and if you see the Moderna success rate, and if you see the protective rate of patients in India from COVID 19, it is almost comparable. So, virus is not really very dangerous in terms of our good immune system. But then, if the people have already contracted COVID, so they were thinking that they will not recontract the COVID again. But if the new viral strain is coming with like different antigens, then there are chances of reinfection. In fact, some doctors they have countered the virus again, who are working with COVID 19 patients. So one may get it, but the chances are low.
Okay, ma'am, thank you. And ma'am, the next question is COVID-19 has also been directly linked to severe neurological effects such as depression and anxiety disorders. What are your views on it?
See, neurological problems are one side, and depression is at the other side. See, COVID can enter the brain and can do a lot of damage to the brain, right? Leading to a lot of neurological disorder. Depression directly may not be the result of the COVID, but maybe due to the conditions imposed because of COVID. Maybe the jobs are gone, the stress is there, the family problems are there. So depression not directly linked to COVID infection. People who are not infected with COVID can have depression too during this lockdown period that we have heard. But COVID, yes, can have a lot of neurological problems, and it can even breach the blood-brain barrier, so it can enter the brain and damage the neurons.
Yes, ma'am.
Um, I
Then,
I want to ask a question, and it was a nice talk. It was a nice recap of everything together, put together. Varsha, I was just thinking that, like a major problem with COVID is the inflammatory side effect, right? And you mentioned that, using IL-6 receptor targeting would be a good therapeutic use. So, I mean, what is the update on that? I would like to really know on that, what is your take that, because, this is going to be effective in different strains of COVID as well, because, we are not targeting the virus at all. We are actually trying to boost the immune system.
Yes, see, there are several aspects to this. One is not every patient will have the same level of cytokine storm as we talked about. Like in India, if you see India, and we are not talking about any other country, but if you see India, the patients actually who are infected with COVID, not all of them are having, lot of issues with the COVID. Majority of them, they are asymptomatic, but some of them really have very severe symptoms. Even the comorbid patients, they don't have, that kind of severe infection. So they are obviously not a good candidate for any kind of anti-inflammatory therapy. But patients who are experiencing high inflammatory reactions, they may be a suitable candidate. But then at what time this therapy has to be given, because these therapies are also expensive ones. So at what times these therapies are to be given, that is very important in the case of COVID-19, because once it starts the damage and it already does its effects on the body, you cannot control that. That is the biggest side effect, because multiple organ failure sometimes is occurring in the patients who are not even presenting the symptoms, extreme symptoms of COVID. So in that condition, it is very difficult to decide which patient should be given this therapy and which patient should not be given this therapy. So that is very difficult to predict for COVID-19, because like in everyday my husband being the clinician, we are every day getting one or the other news of one or two clinicians deaths because of COVID-19. So people don't really know. And one of the doctor, he had fluid lungs. The lungs were all gone. in Madhya Pradesh. So it's very difficult to decide at which stage, because if the viral load is very, very high, then none of the drugs, I think, they will be effective, including this IL-6 antagonist. Hello.
Yes, ma'am. Thank you for the answer, ma'am. This was really informative.
Varsha, I had a question for you. So, what are the steps that are being taken in India for manufacturing of the vaccine to make sure that the supply is there? Are people
Uh,
thinking along those lines?
Yes, like this Serum Institute of India, nearly they have trying to manufacture this 16 crore for 16 crore people. But because
Mm.
of the setback which they experienced in the month of September in the UK trial, so that has not really granted approval. But like in the hospitals and all, cold chains are being maintained by the government. They are maintaining the cold chains everywhere, so that as soon as the vaccine comes, they can supply, and government has made the priority list to whom they will supply the vaccine first. And I think even in the villages, at the village level also, they are maintaining the cold chain, so they are just waiting. As soon as the vaccine will come, I think they will distribute that on the basis of the priority. Obviously, they are doing that,
Mm.
they are working on it. But we are still waiting for Serum Institute of India vaccine to get confirmation and the approval, so that we can have ample amount of vaccine from them. But right now, the Pfizer is the only company whose vaccine has been the UK being the first one, and I think Singapore has also approved Pfizer's vaccine,
Yeah, and
and many more
Moderna is
countries will
coming.
follow. Moderna
Moderna
is
is
coming. Moderna is
yeah.
also coming. Just I think Pfizer and Moderna will be the first ones.